So today we're talking about P450 enzymes and their role in resistance. So it's a two-pronged attack. Both Todd and Roberto have done a lot of research in P450s over the year. Todd is now an assistant professor at Colorado State University. Todd was a researcher at AHRI for a number of years and moved back to the US to take up the position that he's now in. Roberto is one of our AHRI research fellows and has done also some interesting practical work with P450. So I think Todd's going to tell us a little bit about what they are and why they're so difficult to study and what we currently know. And then we'll hand over to Roberto, and Roberto's going to talk a bit about what we know with some specific herbicides. So we'll get the ball rolling. So Todd, over to you. It's afternoon in the US and big storm on the way, and you've been researching P450s for a long time, so I'll hand over to you, Todd, and you can let us know what, you can take it from there, and then we'll stop periodically for questions as we go.
Sure, Peter, thanks. Please let me know if you have any troubles with the audio or the slides, and thanks to you, Peter, and to Lisa for organizing this and inviting me. I'm of course, a proud alum of AHRI, and I'm very happy to have the chance this afternoon and morning to share what we've been up to in researching P450s in plants with the audience. And it's also a pleasure to get to share this webinar with Roberto. Of course, he may be faster than me and when it comes to running and maybe writing papers, but I think we can both chip in a lot on the P450s this morning. So the topic, of course, is understanding P450s, and I just want to back this out a little bit to a higher level and talk about resistance mechanisms, or how does a plant survive a herbicide. When we are talking about weeds that are resistant, we kind of group these into two areas, and what we're talking about this morning falls under what's called non-target site resistance. Every herbicide interacts with some type of protein in the plant, and that's called the target site. And these non-target site mechanisms are anything that slows down or reduces the amount of herbicide that actually reaches the target site in the plant. In particular, today we're talking about enhanced metabolism. It's the ability of the plant to break down the herbicide and reduce how much of it actually reaches the target site. Of course, there are other mechanisms as well that are out there, and they can combine with metabolism, but we're not talking about them today. This would be things like altered translocation, reduced uptake of the herbicide into the leaves, some other things, as well as target site, of course, is an important group of mechanisms. You can have mutations in the target site gene, or, as Roberto knows, one of my personal favorites, gene amplification of the target site gene. But we, again, aren't talking about those today. We're really just talking about metabolism. I do want to point out that even though we're talking about metabolism, all of these mechanisms can combine together. They're not mutually exclusive by any means, which sometimes can further complicate their study. Just to give you an idea of what this looks like in the field, we have some great data from Bayer Crop Science and Dr. Roland Baffa showing some surveys of the extent of target site and what we call metabolic resistance in blackgrass in England. And what you're looking at here is a number of sites where the majority of the individuals in the population either had target site, these red dots, or metabolic resistance, the yellow dots. As you can see, a lot of the blackgrass in the UK and the wheat growing regions are these darker blue ones. Obviously not a lot of wheat production down here around London, but as you get further north, these generally have target site resistance mechanisms in dominating, but certainly a few populations with metabolic resistance. And the take-home message is that target site resistance is specific to that mode of action. So this is for the group 1 ACCase inhibitors, and a target site mechanism will only make it resistant to other ACCase inhibitors, sometimes specific to a certain chemical family, but not to, for example, group B herbicides or other modes of action. We have a different situation, however, some survey data from wheat growing regions in France, And you can see that the majority, 3/4 of the populations, are the yellow dots. So they have metabolic resistance. That is, they break down the ACCase herbicide quite quickly, and that's what allows them to survive. And they don't have target site mutations, whereas about 1/4 of the populations have a majority of target site. The difference and why we care so much about metabolic resistance in P450s is that this enhanced metabolism can confer resistance across different modes of action. So say, for example, if these populations were first selected with group A herbicides, the ACCase inhibitors, switching to group B herbicides, you might find that there's already some resistance out there because of this enhanced metabolism that doesn't happen with target site. And again, I'd just like to emphasize that these two mechanisms can and do co-occur. Now, what is it in particular about metabolism-based resistance that we care so much about, and why is it difficult to study? Generally, what we're looking at here is a change in the expression of a gene that lets the plant metabolize the herbicide. And specifically, a type of gene called cytochrome P450 monooxygenase is the gene that we're talking about, or P450s for short. These are a very common type of gene in plants. Animals have these as well. And they're enzymes that perform types of reactions known as hydroxylations or redox. And they're very important in plants for their function as biochemical factories. If you think of plants, they make lots of secondary compounds. Some of our favorites that we might encounter in our daily life, such as caffeine or nicotine or some other of those type of compounds, these are things that plants make in which they use cytochrome P450s to make them. So they're just common genes in plants, and they also sometimes have the function to detoxify xenobiotics, say something a predatory insect might inject into the plant, or a fungus. They can help the plant break those down. And it's a change in the expression of those genes that actually might confer the resistance. So the plant might normally have a very low level of expression, but you can select for individuals that have a much higher level of expression, and that could then allow that plant to break down the herbicide in time to survive. Again, why is this important? It's because these types of genes might actually provide resistance to herbicides from different modes of action, and that sort of complicates our typical recommendations for managing resistance, because rotations and even mixtures might not have the same effect in managing that we would normally predict. Another big trouble is that this type of metabolic resistance from P450s could even confer resistance to herbicides that haven't yet been discovered or introduced to the market. You can think of this as a generalist or a broad spectrum adaptive response. So the plant is sort of gearing up to bring down all challengers. It's sort of like your all-rounder in cricket, just able to handle anything that comes its way and do a good job with it. Do you like the cricket reference, Pete? I thought about that for a while.
Yeah, well done.
Okay. Now, one of the ways we study these P450s is to do what's known as whole genome sequencing or whole transcriptome sequencing. And what I'd like to tell everyone is that the costs for this have come way down. It's really interesting. In just 10 years ago, in 2007, it cost about $10 million to get the amount of sequence data equal to the human genome. And now that costs well less than $10,000. In fact, you could probably get that much sequence data for about $1,000 now. So just a huge reduction in cost. Certainly very few other things in the world have come down in cost that much in 10 years. So we can do the sequencing, and why do we need to do that again? P450s are a big pain to study. That's what I want everyone to have as the take-home message. This is the metabolism pathway for the herbicide diclofop in wheat. So this is really well known. You start out with diclofop methyl, and then it gets demethylated by an esterase, and then you get this diclofop acid. This is the herbicide. But here, cytochrome P450 can perform a hydroxylation right here. I think I even have a nice pointer I can use. Yeah, right here you see there's a hydroxylation that happens. Now this is no longer toxic. So you say, okay, that's a P450. Well, the model plant Arabidopsis has more than 300 gene models for P450s. So it's very hard to just know, well, okay, it's a P450. There are over 300 different ones. So how do you know which one it is? And then there's a second step here where glucose gets conjugated. Well, Arabidopsis has more than 250 gene models for this type of gene, glucosyltransferase. And that's what makes it complicated, and that's why we want to use next generation sequencing to look at the expression of all of these genes at once. I'd like to give you an example of a study that we did with ryegrass from Australia. A lot of the audience might be familiar with this work. This is something that Paul Neve did when he was working with Steve Powell's. He did recurrent selection with diclofop over several generations and was actually able to select for a 40-fold increase in resistance to the herbicide diclofop from the original susceptible population. No, he didn't select any of those target site mutations in the ACCH gene, but he did select for enhanced diclofop metabolism. And we wanted to understand what we thought it would be P450s involved, and we wanted to see what those are. So I mentioned the next generation sequencing. This is what it actually looks like when you get a hold of it in a computer. You've got different gene models that you assemble, and this linear sequence left to right is the actual sequence of the gene. So, you know, the A, C, T, G, C, etc. that actually makes up the codons for the amino acids. This is a cytochrome P450 known as 72A, and you notice that there are lots of little black lines under this one for resistant and very few black lines under this one for susceptible. And that's what makes the difference. This resistant individual, it has a much higher expression of this gene. You can kind of think of it as the dimmer switch on a light bulb in a house. You know, you might install a light switch that's just simply on or off, and it gives you one level of light. Or you can go for that dimmer switch that lets you, you know, go for that kind of laid back, a little bit more casual feel with a lower light, or the interrogation mode with the super bright light. And that's basically how plants can control their genes. They have them on something like a dimmer switch. And this resistant individual is making a lot of RNA for this P450, so it makes a lot more of the protein. The susceptible individual also makes it, but it makes a lot less.
Oh, you know I love the dimmer switch analogy, Todd.
I know, that's yeah. Go ahead.
It's one of the great ones. Hey, just with that graph there, so just above the line and below, so below I'm getting is about the level of expression, but the chart above the line there, what is that?
That is just letting you visualize along the length of this gene how many counts do you have at any given point. So this y-axis is how many you actually count how many reads are present under the curve at this exact spot or at this spot. So you notice here where the y-axis is higher. Can you see my pointer when I'm doing this?
Yep. Yeah, it's fine.
So you see this is the highest. This is where the most reads are stacked up underneath it. So that's why what we actually measure to do the statistics on is you take the sum of all of these little lines, and that gives you a number. And for example, the number for this one might be about 400 counts, and for this one might be about 20 or 30. It's a big difference.
Right.
But this is just letting you sort of visualize it and go, oh, okay, there's a lot going on here, not much here. You could also inspect this for any unusual patterns. You know, if it just totally disappeared in one spot and came back, that might suggest that you have something going on with the RNA that you need to look into a little bit more. But typically, you don't see that.
Yeah, that's great. And everyone listening in out there, if any questions come up, make sure you type them into that question box, and we'll take some questions periodically. Thanks, Todd.
Sure. I think just a bit more here from my end. This is how we can actually translate this information into something useful, and that is where we'd like to go with this is basically to walk out into someone's paddock and walk up to a ryegrass plant and just when it's come up, or even collect some seeds and germinate them over the say the summer, so we can get a good diagnostic of resistance. But as quickly as we can, move from sampling a leaf to say, yes, you've got metabolic resistance to group A and group B herbicides. No, you don't. That's where we'd like to go. We're not there. But what we are able to do with this information is put together several different genes that sort of form a diagnostic panel. Think of it like your markers in your blood for, say, hypertension or heart disease. If you have one or two of those markers that are elevated, your doctor might say, you need to cut back on the Vegemite a little and the meat pies a little bit, but for the most part, you're fine. Or if several of them are high, your doctor might say, you know what, you need to get out on the track a little bit more, you need to spend some more time walking, make some more substantial changes. And that's what we're trying to do here. We found a suite of 4 or 5 different genes that together we think give us a good picture of being able to diagnose metabolic resistance due to P450s. But it's certainly ongoing, and I know the group in AHRI, they've got some great work going right now to really pin this down around which P450s we should look at for this diagnostic panel, so that we can give people good advice as quickly as we can. And so then that's that turns it over to Roberto. I guess I would like to ask Pete if anybody's got any questions there, or again, why do we care about P450s? What are they? What do we do with this information?
Okay, thanks, Todd. And yeah, that's great. So what I'm getting from that, now correct me if I'm wrong, that yeah, P450s are obviously important for resistance, but extremely difficult to study because we've got multiple P450s and multiple genes for each P450. Is that right?
Well, that's right. P450 is a very big group of enzymes in the plant, and they have a very diverse function. We call them all P450s, but in fact they're very different from each other. But they all do something like what I showed. They can take one of those aromatic rings that's in a compound, such as a herbicide, and hydroxylate it, or do different things to modify that ring structure. That's essentially what they have in common. And a whole lot of them don't do anything to herbicides, but some of them do. It's also really hard to isolate the P450 protein from a plant such as ryegrass because they don't make very much of it, and it's also bound to an organelle in the cell called the endoplasmic reticulum. And what that means for everybody listening is that it's just really a pain to get that activity out of an extraction and test it in the lab. So we're hopeful that this approach of actually looking at the gene sequence helps us identify which ones are doing that. Then we can study the function of that very specific, such as I showed, a cytochrome P450 72A number 1. There's a whole numbering system for these P450s to help you identify which one is which.
Yeah, and if we did understand them more, is it potential that we might be able to develop new herbicides or maybe learn to switch them off? Or, what would potentially, you mentioned a test, but what's some of the other end-game benefits that we could do once we understand them more?
Well, I think that segues really well into what Roberto is going to talk about. If we understand that this is how the weeds are resistant to the herbicide, we can do some things chemically to try to inhibit these P450s so they won't work on the herbicide, or you've probably had some communication with people around RNAi or RNA interference, and that would be a great application for this as well, because if you know the sequence of the specific P450 and you had an RNAi system, you could silence that specific P450 so the plant would presumably then be susceptible to the herbicide again.
Excellent. Now we do have a couple of questions come in, Todd, so Roberto, don't just hold your breath for a minute. We'll get to you, mate. But good question come in. If P450 is high in ryegrass, how important are they in wild radish? So is there a difference between the grass plants and the broadleaf plants in terms of how many they have and how active they are?
Yeah. In terms of the actual number of total P450s, it's fairly similar between grasses and broadleaves, such as radish. We know more about P450s in grasses because people have been studying this metabolic resistance for a longer time in grasses. I think Steve has some papers from his group from sort of the early and mid-90s on metabolic resistance in ryegrass. So it's been studied for longer. I think now researchers in different areas are starting to look at metabolic resistance in broadleaves and are starting to find some examples of it. So it probably still is important. We just don't have as much information on how important it is. Partly this is also due because there are a lot of selective herbicides used in wheat that are selective because wheat metabolizes them. And then, as you know, ryegrass is also a grass, so it presumably can also metabolize these herbicides. It just doesn't do it as quickly. And with enough selection pressure, we've kind of selected for a wheat-like mechanism in ryegrass. Whereas many of the herbicides used to control wild radish are not necessarily selective in the same way, grass within grass. You don't have so many selective herbicides that would control wild radish in a broadleaf crop, for example. But there are some cases now. One good one would be for triazine resistance. Some cases of metabolic resistance have been found with a GST, a gene that Roberto is going to talk about here. And I'd say it's an area that we're just starting to get insights into now.
Thanks, Todd. Okay, I've got more questions. What cell function do the various P450s have in the absence of herbicides? I think you've answered this a little bit already, but do they all do something similar, or are they involved in just a whole range of functions?
They're in a whole range of functions. As I said, they make so many different compounds in the plants, from everything from lignans and phenols, and so components of the cell wall. Lots of reactions there. Lots of protective compounds. They're involved in synthesizing chlorophyll. Just about anything that in the plant that would have a 6-carbon ring on it, they're probably involved with at some point. Some of them just sort of serendipitously have the ability to detoxify foreign compounds that come into the plant. And so some of them have evolved to detoxify some, say, a toxin a fungus might inject into the plant or an insect. And then some of them just happen to have activity on herbicides. And that's what we don't understand yet. What is it about their protein structure and the chemical structure of herbicides that allows the P450s to have this activity? I think that's our research frontier right now.
Yeah. I've got more questions, Todd, so keep going. So we know there's metabolic cross-resistance in Australia between ACCAs and flamprop. Do we know if the same P450s metabolize ACCAs and ALS, or is there metabolic cross-resistance, or are these two groups degraded by different P450s?
I would say we can't quite say for sure on that one yet. Certainly, I think AHRI has some research that I would say within the next year, Pete, you'll be able to write a really nice AHRI newsletter on that one, because I think they'll be able to answer this exact question. They're working on it. But right now, I think there are two scenarios. There could be a single P450 that metabolizes group A and group B. That's possible. Or I mentioned the dimmer switch. That's called a promoter of the gene, and that's what signals the gene to turn on. Those are controlled by things called transcription factors. It's possible that when we use a group A herbicide and then switch to a group B herbicide, we could actually be selecting for a different transcription factor that really is functioning as that dimmer switch, and that transcription factor might work on 4 or 5 different P450s, and so one of them handles the group A, a different one handles the group B. But when you test the whole plant, you see the same effect, that it's resistant to both. But you don't know that it's two different genes. But again, I'd say that's really a watch this space. We should have a good answer pretty soon.
Okay. Another one here. How many seasons does it typically take for these resistant populations to be apparent in the field?
You know, that might be a good one for Roberto in terms of he's got some surveys here. Yeah, I'll just, am I allowed one handball, Pete?
Yeah, you can handball that to Roberto.
Okay.
Roberto.
Okay, is it my turn? Good. I would have kept listening to Todd for the whole morning. Definitely Todd is one of my favorite scientists. Let's say just behind Steve Powers and Chris Preston, it would be probably third ranked. So Todd, well done. You get a bronze medal today. So yeah, my role here is well, first of all, we had such a comprehensive view, and Todd was great in making things simple. So whoever wasn't born a molecular biologist could really understand the dynamics and all the complexities involved in this non-target side resistance P450 based. My talk today and my bit is just to basically having an Australian focus and bring on this case study that we did on ryegrass. And only just to simplify to the extreme, I will only add one more thing. So no big deal. Hopefully everything will be clear. And I start here with a graph showing the increase of multiple resistance in ryegrass that was assessed through random surveys done in Western Australia. That is what WA stands for. And if you can see the red bar, it has inevitably increased. So we went from 50% multiple resistance in the late 90s to basically 98% in 2010 when the last random survey was done by Michelle Owen. And if we look at the geographical scale, we can see the Western Australian wheat belt that is covered by those red dots. Basically, you will only able to see one green dot there, and that means that basically most of the ryegrass that grows in cropped paddocks is multiple resistant, and it is multiple resistant to post-emergence herbicide. And Todd was talking before about the metabolic versus target site resistance. Well, a study in those populations there in ryegrass shown that basically 70% of them have both, and that is very much in tune with the cross-pollinated nature of annual ryegrass that basically is able to stack those traits in single plants. And that is why basically while resistance was increasing so much, farmers started to rely more and more heavily on pre-emergent herbicide. And I put here this slide, wanted to basically highlight two concepts. First of all, those pre-em herbicides have been discovered sometimes more than 50 years ago. And I put in brackets the mode of action of those 5 different herbicides that are basically the bottom line, the one that are adopted mostly by Australian farmers. And you can see that there are only 2 modes of action that we can rotate with. So it is great that all of them are doing a great job, but again, there is little diversity already. And we are relying on them quite heavily. And we definitely don't want having a bar like the red bar showing resistance to those ones. So it was time to act. It has been time to act, and still now all the ongoing efforts is just to support and make sure that those herbicides will last for a long time. The last one to come to the market has been Pyroxasulfone. It was commercialized in 2012. And well before that, we knew that it would have been a very good herbicide tool coming to the Australian and also to the global market. And for the first time, and then the audience will correct me if I'm wrong, but we did a study that was unique because we wanted to assess the risk of resistance to this new potential tool. Before this herbicide was released commercially, we wanted to see whether resistance could have evolved in ryegrass. Todd mentioned already before that P450 genes and all these non-target side metabolic resistance genes are so dangerous because they can endow, confer resistance to something that has not been discovered yet or has not hit the market yet. And so this is exactly what we wanted to understand, and we did a study, we did a selection here in the University of WA in controlled conditions with pot-grown plants, and we shown, and this may, Peter, partially answer that question that came in before, how many seasons it would take for resistance to evolve. Well, if you don't have diversity, even you can break a great herbicide such as Pyroxasulfone in a very short time, because basically what we wanted to show here was that in 3 generations we had a tenfold shift in resistance. So we had tenfold resistance evolving after only 3 applications, 3 generations of Pyroxasulfone used at low rates. And so then we also assessed, and we didn't expect that much, was that because of selection with Pyroxasulfone, we had cross-resistance evolving to those 2 thiocarbamates herbicide, and one was prosulfocarb, and the other was trialate. So basically, from the 5 herbicides that I shown initially, 4 of them would have been lost already. So what to do? We were a bit struck by this result, but what we did was trying to study the mechanism of resistance, try to get maximum insights in how we could possibly have managed or delayed resistance at the maximum level. And we tried a few known P450 inhibitors, and yeah, most of them didn't work. So we could not even in the lab, in the glass house, reverse resistance. And then we came across to this soil applied insecticide, an organophosphate insecticide. So that, because it was a granular soil applied, could have had some soil activity, could have interacted with the seed along with a pre-em herbicide. And so basically what we did, we applied this insecticide to the resistant seed, and then after having applied this insecticide, we sprayed the normal pre-emergent herbicide, and we then observed what happened. And I will show you here a couple of examples of how this organophosphate insecticide has interacted with 2 important herbicides. One is Trifluralin, and so we should start to read this photo from the far right-hand side. So basically, we applied a given rate of Trifluralin. That rate was able to control S plant, meaning was able to kill all the susceptible plants. The total opposite was at the far left-hand side, where the given rate of Trifluralin did not kill resistant ryegrass. But when we applied the mix of Trifluralin and Phorate, this organophosphate insecticide, we had a great reversal of resistance. So basically, we could kill a resistant population. So we found, we believed we had a cure for everything, and so immediately we tried that with Pyroxasulfone. Sakura is the commercial name in Australia, and I guess Zidua is in the US, and so we put it together, and again the dose we use in this particular pot study was good. It could control the susceptible population. It couldn't control those P6 at the far left. It couldn't control the resistant population, the one that we obtained by recurrently selecting at low rates, as I showed before. But we could have some level of interaction between the two, not a great result as we had with Trifluralin. And basically, we will try to understand in the next few slides that I'm going to show why that has happened, why there was no full reversal of resistance. And again, just I mentioned before, but basically, we only found so far one compound that really gave us good interaction with the herbicides, and we tried all the other well-known foliar applied or hydroponic applied P450 inhibitors, but none of them was synergizing with pyroxasulfone. They are known to synergize with ACCA, Group A, or ALS, Group B herbicides, but none of them work with pyroxasulfone. If there is any question coming in, Pete, please stop me anytime you want, otherwise I keep going.
Well, we've got a pile of questions. We will, I think we'll let you keep going. It will be 5 minutes and then we'll finish off with a heap of
Okay.
I thought Todd's question answers were long, but I know you're Italian, but that was ridiculous. That was about a 10-minute answer to a question. But it's all good. Great job.
Yes. Thanks, Pete. I keep on going then. So just this graph to summarize what I've just shown in photos. This is when we try to quantify this reversal, and we express that reversal as a percent of the lethal dose for the 50% of the population, of the resistant population. And you can clearly see, if you look at the blue bar, the best results we got was with Trifluralin. The blue bar is the biggest, and only left a tiny bit of red that is basically representing resistance. I wanted to put in context with Group B herbicides. It is well known that those insecticides well synergize with Group B's herbicide, but again, the job was not as good as with Trifluralin. And the bottom bar is the results with pyroxasulfone. Again, we only had 40%, 40-45% reversal, but still there was quite a big chunk of resistance that was still there, and we could not manage to kill those plants. So why is that? Why the P450 inhibitor didn't do the job completely? And this is the one more thing that I wanted to add on Todd's introduction. Basically, we went on and studied the resistance mechanism for pyroxasulfone resistance, and we have here evidence that it is metabolic resistance. So here are data showing the fate, what happens to pyroxasulfone in susceptible and resistant plants. And you can see, if you look at the orange bar, that the orange bar, pyroxasulfone, is much, much lower, even 1 day after the treatment, is much lower in the resistant plants. So that is exactly what happens in the crop, exactly what happens in wheat plants. Pyroxasulfone is metabolized, and the metabolites are all those blue, red, and green bars above the orange bit. So in resistant ryegrass, it happens much faster, more rapid decrease of C14 labeled pyroxasulfone. And again, we went a step beyond, because we wanted to understand what those metabolites were. And with mass spectrometry, we were able to basically, in conjunction with Kumiai researchers that invented and discovered this herbicide, we basically worked out what those metabolites are, the chemical structure of them. And basically, when you have that and you have the chemical structure of the parent, pyroxasulfone, then you can speculate, you can basically understand what is the mechanism behind. And basically, The mechanism behind involves not P450 anymore, but involves also GST. GST, again, another super family of enzymes. They don't just hydroxylate the molecule, but they are good in basically attaching a tiny protein, a tiny tripeptide to the herbicide, so that the herbicide is inactive, is detoxified. And this is exactly what this quite complex pathway shows, that there were metabolites formed. There was pyroxasulfone up there on the on the left-hand side, and GST basically was this enzyme believed to have allowed metabolism of pyroxasulfone, metabolism of Sakura.
So Roberto, if I can just interject quickly, GSTs, we also put them in the metabolic resistance category, don't we? Is that right?
Yes, it is exactly right, and they are fewer. There is a smaller number of them in plants. Todd mentioned this 300 in Arabidopsis. We would say that GST is probably around 50, but still they are as complex as the P450. So not an easier job to find them and understand how they are regulated, how they are expressed. So again, another quite big hurdle to overcome. Hopefully, the genomics has the potential to, you know, unravel them, discover them, and study a bit further. And we wanted to again to look at whether we had some molecular confirmation of what we have just seen here. And so basically, we searched the literature. We found a bunch of GST genes or GST contigs that had been identified with transcriptomics, with these genomics, things that Todd was talking before, and we tried them on our resistant plants. And basically, we found that, you know, phenotypic resistance, pyroxasulfone resistance, was highly correlated to the expression of a couple of those GST. You can see the black bars there matching, meaning that basically we had, we believe we can say that GST is highly expressed in our plants, and that is why those plants are able to basically survive pyroxasulfone application. And basically this is my last slide, trying to bring all back into some practical recommendations, try to see whether we can bring all this deep and basic science into the applied world. So here, Peter, is the well-known boxes graph, the recommendation that a couple of years ago you and myself worked out as the message for the agronomists. And basically, there are two things that are important here. One was the emphasis that we keep having on using the full herbicide dose, the full rate. We want to use those herbicides at the maximum power. And the other is the rotation, the improved rotation pattern that we wanted to come up with as the best rotation possible that we can basically do in the field with the information that we have already collected in basically 10 years of research. So we put propyzamide on its own, and we did that because basically there is no resistance that has evolved to this herbicide. We put in the same box pyroxasulfone, Sakura, prosulfocarb, Boxer Gold, and Trialate in one box only, because we have seen, as I shown, cross-resistance evolving to these herbicides under pyroxasulfone selection. And I'm sure there will be questions on this and why we did that, and the fact that, you know, there are already exceptions to the rule. This is not just, you know, we are dealing with biological system, and so the exception is actually the rule. we expect diversity occurring, but this was what our data were showing. There was cross-resistance between those herbicides, and so we wanted to put them in the same box because rotation between them may not be as effective as rotation between the boxes. And then we put Trifluralin on the left because at that time we believed that Trifluralin resistance was target side. But today we have discovered basically that is in this population it is metabolism-based, and see whether this is still true. And I just put here what likely the mechanism will be, and everything still is applicable and remains basically the same. I believe that we can say that most likely GST may explain resistance to pyroxasulfone, prosulfocarbon trialate. We found that the application of the insecticide and known P450 inhibitors was reversing Trifluralin resistance. And so I guess that this still is a great message, and the agronomists should be aware of it. And well, there may be questions coming in about mixtures. Reality is that we don't have yet good data on how mixtures can delay resistance. We have lots of data, I'm sure, coming from the field about the improved control of ryegrass, but there is not yet much data on how mixtures may delay resistance, and this will be certainly my focus for the next few years of research.
Thanks, Roberto. You've done a great job, and look, I think this is a great story between the two of you, Todd and Roberto. So, yeah, it really is showing that there's been this era of understanding this metabolic resistance, and now, Roberto, you've put it into practice in pots, and that's turning into a strong recommendation like you have on the screen there. So it's a good story, and we've got a pile of questions, and we've got 10 minutes. So I know that, Roberto, you are Italian, and that Todd, you are a scientist who has long explanations of these questions. But I think we give some nice short answers. We'll get through a few of these, guys. So, look, I think this next one is for Todd. The question is, and I think this is right up your alley, Todd. So the question is, in a resistant plant, P450 does not necessarily break down the herbicide. It causes overexpression of the gene so the plant can bypass the toxicity of the herbicide. Is this right? And I think this is a bit of possibly a confusion between P450s and gene amplification, Todd. So maybe you might explain the difference.
Yeah, the P450 is the gene, and it is the gene that has increased expression. It could be due to gene amplification, or it could be due to this dimmer switch idea, but the P450 is the gene that does the job of breaking down the herbicide. If you can get 10 times more P450, you can break down the herbicide maybe 10 times faster and survive.
Great answer. So gene amplification or overexpression, or even a bit of both.
Yeah, a bit of both.
So the next one is, what is the metabolism enzyme difference in ryegrass for the germinating seed, emerging seedling, and young seedling? So I don't know if we know the answer to this, but is there a difference in the levels of P450s and GSTs and so on through the life of the plant?
It's a great question, and I think whoever asked it should contact me about working on a PhD. That's definitely one thing we're looking at right now. Sometimes a great example of this is resistance to PPO and HBPD herbicides in the U.S. We have populations of pigweed that are resistant post-emergent but not pre-emergent. Why is that? Is it because they overexpress the P450 as a seedling but not as a germinating seed or in the root? We just don't know right now, but that's definitely an active area of research. It certainly could be the case. These are regulated throughout the life of the plant and in different tissues, so they definitely could have different regulation.
Right, great. The guy that asked that question, I'm not going to name him, but He is a good scientist, and he is nearing the end of his career, I would say, Todd. So not sure if he's ready to take on a PhD. Maybe he will, but yeah, good question, and thanks for that. So the next one, I'm not sure if I understand it, and Roberto, if you could flick up to slide 10, please. Does the resistant population in slide 10 always, well, that's going to be a bit, anyway, does the resistant population in slide 10 always express upregulation of that specific P450, or does it become upregulated after the application of herbicide?
Well, basically, the question is about constitutive versus induced P450 activity. I guess that induction is definitely an important one. We don't know yet which plays the most important role, and Todd may then come into help here. What we could see in our GST overexpressed plants was that that was constitutive, because basically we just simply assess the expression level of GST in treated versus untreated plants, and that was the same, didn't change. So we very much believe that those GST were turned on in those plants, and that is why they were basically dealing with the herbicide. Todd, if you want to add something about this induction versus constitutive, I'm happy to do it for you.
Yeah, I would just say that I think that's an area we're still very actively looking at. Either one is plausible. It could happen. Most of the evidence so far suggests these things are probably constitutive, but we could always find a case where they're induced.
Okay, constitutive is a big word. Can you give me the meaning of that, Todd?
I should remember. Probably we're not allowed to use big words in the U.S. anymore with our Twitter president. No, okay, I had to make one joke. Constitutive means all the time. It's on. It's not responding to environmental cues or signals. It's just on. And so it's like the on-off switch that's just somebody taped it into the on position.
Thank you for taking it back to the dimmer switch. I understand now, and please excuse my lack of vocab. Okay, another one from one of our Western Australian agronomists. In ryegrass, how many P450 types have been found to be active on the herbicides? And I think you had that answer there for blackgrass. So Todd, this might be for you. I think you did a study showing how many different P450s were active. Is that right?
Yeah, we don't have a certain answer to this yet, but I would say, I would guess it's in the range of 4 to 6. P450s probably have some kind of role in acting on herbicides, and there may be some that are the most important and some that contribute a little bit. But in most of the studies so far in ryegrass and blackgrass, they're typically identifying 3 or 4 different P450s, I would say, that might have a role.
Yeah, great answer. Okay, so Roberto, this is one from specifically from your research. What were the low rates of pyroxasulfone in terms of active ingredient grams in these experiments compared to the label rate, and are these rates practiced in the field?
Well, the rate I used to select the first progeny was 60 grams of active ingredient. So 60% of the full rate, which is 100 grams of active. I don't think that there are many farmers that still use low rates, and we have definitely shown that it's something that should be avoided, because you are just putting yourself into a greater risk. First of all, your weed control may not be as good, and also you are running the risk of evolving this creeping or polygenic resistance. That is something that you don't want. You want just to try to maximize your kill rate and then lower your numbers, and then maybe coming back at the end of the season and practice some non-chemical weed control tools such as harvest with seed control.
So Roberto, from memory from your research, it was a 60% rate for the first generation and then a full rate from then on, is that right?
Correct. We actually went beyond the full rate because we had witnessed already a level of resistance evolving, and so we wanted to keep the selection intensity consistent across generations, and also yes, we started back in 2006 and we wanted to maximize the outcome, so we put on a strong selection intensity in the subsequent generations. And when Todd was here, we actually selected the highest level of resistance by applying 240 grams of active. So telling you two things, that those plants were highly resistant and resistant as the wheat crop would be, and yes, and basically we obtained a population that is highly resistant and quite homogeneous, and so a great resource to then do subsequent studies at the molecular level that are always complex, always too difficult. And so we wanted also to, apart from proving a concept, also having some good material to do basic science on and avoiding variability.
Yeah. Now we must point out you did start with a nasty ryegrass population to start with, but I guess, and this is following up with some other questions that have come through for some people here, I guess the concern for me there is with a herbicide like any of the pre-emergence, you apply them and then the rate degrades through time. So if we have a late germinating ryegrass, it gets a lower dose. And in your work, you had one low dose and then high doses from then on. So it does just show how fragile these herbicides could potentially be, that we could get a late germinating weed, get one low dose, and that one event could be enough to select for resistant survivors. So have I got that right, Roberto, or am I overstating that a little bit?
Well, you definitely have a line of interpretation, and basically, in my opinion, what we don't have is a good, solid set of data that show, you know, what is the effect of a degrading herbicide and what does that do for resistance to evolve. I'm actually at the moment doing that study funded by GRDC, and so hopefully I will be able to, again, to at the very least put some insights into this, because one thing is, yeah, if you have a herbicide that doesn't degrade in the soil and is always the rate is always high, you have basically continued selection, and so that in a way could actually speed up resistance evolution, and this appears to be, for example, the example of sulfonylureas in alkaline soils, where one application was able to select for resistance because that herbicide lasted 3 years in the soil at a very, and basically the selection intensity was very high. Vice versa, you have this low dose data that show that when the rate decreases, then you have, you know, a low selection intensity that allows many more individuals to escape, and those can cross-pollinate and basically start these creeping resistance type of processes. And so both are true. Is not easy. Definitely, it shows that once again, if you rely on herbicides only, you may have a greater risk of running into problems. And so again, we try to back up as much as we can with scientific, empirical data, but the reality is always one, that any herbicide you will use, more or less robust, you will select for resistance, and so you need to be very well aware of it. And we chose one of the best herbicides brought into the market, such as Pyroxasulfone, and when we broke it in 3 years. So again, then we can speculate on all the miniaturistic and small details, but the reality is that just use as much diversity as you can and use non-chemical tools.
That's it. Thanks, Roberto. Now listen, everyone, we are at the end of our hour that we advertised, and so we'll understand if you need to leave. But I thought, given that we have these two learned colleagues on the line, we might just let it run for another 5 or 10 minutes to finish off answering your questions. So we understand if you need to leave now, but we do have a few more questions, and so it'd be good to just finish off with them. Roberto, just quickly, in your research, how long before the herbicide application was the organophosphate applied? And there's questions about which organophosphates you were using. I don't know if you left that out deliberately, or and also there's other questions around if we had, you know, why can't we just use organophosphates in the crop? That I think we might kill the crop. You can tell us about that. But yeah, how long before you applied herbicide did you put on the organophosphates?
Well, basically, I had all my pots ready to spray, and I manually applied the organophosphate, and then I sprayed. So it might have been 10 to 15 minutes. And basically, it was amazing. It was amazing that the organophosphate had to go into the solution, and then it was very effective. So that shows that, you know, the ability of the insecticide to block those enzymes was basically as soon as they could get in the seed, germinating seed, and into the cell, then they were active. And the other was, if we can kill the crop. Again, here, you know, to me, the big difference between pre-emergent and post-emergence is that in particular, if we think about an no-till system, we have a great opportunity here to find soil-active synergies and try to. Lots of research should occur now on, because we proved the concept that by applying this mixture, we could kill the ryegrass. I have to say that even in pots, that mixture was not increasing the efficacy of the herbicide on the crop. But in a no-till system where basically the point is removing some of this herbicide from the fur, we could have actually the opportunity to come in with something that would be normally toxic even to the crop, and then removing and having it work in a no-till system. And then, have I answered the question, or?
Yeah. Did you want to disclose which insecticides you were using, or you want to?
Well, this was Phorate. Phorate is not registered in wheat. It is registered in cotton, and I used basically 10 kilos of this insecticide, which is within the label for using cotton. But again, I'm sure that if we look we will find other that may be, you know, even possible to use in wheat, such as other insecticide or from the same class. But that research has just not been done, and I think it should be done.
But am I right in suggesting that it's difficult to find one that if we switch off the P450s in the weeds, we might switch them off in the wheat as well, and the wheat or crop may die? So it's tricky to find a selective.
It is. But with the pre-em, it is better than the post-em. The post-em would just be impossible. You know, malathion plus glean, it would kill ryegrass, metabolic resistant ryegrass, but it would definitely kill the crop. And that would be because we apply in post. But in pre, that could still work.
Okay.
I might add, Peter, the way that we came across Phorate, we had a small grant in AHRI a couple years ago to try to find a wheat selective insecticide to synergize herbicides, because the ones that we had looked at previously did exactly what the questioner is asking about. They knocked out the P450s in the wheat. So we screened a whole range of insecticides and came across Phorate as one of our better candidates to have the potential for this selective P450 reversal.
Excellent. Yeah. So there's potential. There's a line of research there which could help us by understanding P450s and the insecticide interaction. We could potentially use that to come up with a way of switching off resistance in certain weeds and being a tool for the future. Okay, so I think we might just have one more question, then some general comments from you both. The last question for you, Todd. One of the listeners came in a bit late and didn't understand how a herbicide that has not yet been developed already be resistant. So can you just give us that in a nutshell, Todd?
Sure. The way that would happen is if, say a new herbicide is discovered and it's a new mode of action, but it has a chemical feature in the molecule that is in common with a herbicide that's already used in a different mode of action. So we would normally think of these as, great, different mode of action, we can rotate to it using mixtures, but to the P450 or other metabolic enzyme, that herbicide might look exactly the same if it has the same chemical feature that docks into that protein and it can hydroxylate it and inactivate it. It's the metabolism is not based at all on mode of action. It's actually based on chemical structure. So a new herbicide might have some chemical structure in common with our existing herbicides.
Okay, excellent. Well, thank you very much, guys. Now, really just probably a final comment from both of you. We've heard a lot about P450s and your research and so on. Firstly, you, Todd, where do you see the research heading now, and what sort of future benefits could we potentially gain from the research?
I see it going into some of the good questions that were asked today. I have some work going on, and others have work in broadleaf metabolic resistance. I think that's a big area we need to understand, as well as characterizing the developmental timing of these, specifically pre-emergence versus post-emergence P450 activity. But where we'd like to go would be, some of these designer P450 inhibitors. For example, if we know the exact 3D protein structure of the P450, we might be able to design something that works a lot better than Phorate, and then maybe that can be sold with the herbicide so it can work again against metabolic resistance. It could go the genetic route in terms of RNAi, as we talked about. That's certainly a possibility. And it may help us improve our crops so that we can safen our crops using P450s against herbicides that normally wouldn't be selective, and thereby perhaps introduce a new mode of action into a crop. So I think it's all around sort of clever technology and mixtures to help combat this really big problem of metabolic resistance.
Thanks, mate. And Roberto, your future research and where you think that might lead?
Well, it is based on the fact that there has not been much herbicide discovery in the last 20 years. So much of my research is basically to come up with herbicide solution within the already existing chemicals that we have, and try basically to get them work as long as we can and to preserve their efficacy. This is basically what I'm focusing on.
Thanks, Roberto. So thanks. Was there another comment coming in there? No, thanks, Roberto. God, that's been great. I mean, you guys were great mates in AHRI and worked together then, and it's just been fantastic today that we could bring your research back together with one of you on the other side of the planet to where we are. So Todd, thank you very much for finishing your day late and getting home late to your family and joining in with this. It's great we can use technology like this, that we bring researchers together and help understand these things. So thank you, both of you.
Yeah, it really is it. And thanks to you, Peter and Lisa, for putting this on. It is great to be able to join in this kind of discussion despite the time zone difference. Good work.
Yeah, that's great.
And last comment. Yes, thank you, Peter. And Lisa, Peter, is always good to have you as a moderator because you really bring in a very good vibe. And thank you, Todd, to bring in really a great deal of science, and I guess that my comment would be that this is just leading well up to next year